Okay. Coming back off of part 4, I believe. God. Anyways, you know what this is. Perforin attacking, and then the granulocytes go in. Alright, here T cell activation requires binding of a specific antigen. It occurs through antigen presentation, which bridges MHC class 2 of the APC to the TCR of the T cell. And class 2 is going to be with the T helper cell. Initiates a signal cascade which involves other membrane-bound proteins and intracellular messengers, and a second signal is required for lymphocyte proliferation, differentiation, and expression of specific cytokine genes also occurs. And if no second signal occurs, the T cell becomes anergic. I guess that means it's activated, but it could not respond. Lymphocyte proliferation. So yeah, differentiation and expression of specific cytokine genes also occurs. So it's not going to be able to proliferate, differentiate, and express cytokine genes if there is no second signal. So here we have, on the left, we have MHC class 2 from a macrophage or something like that, binding to an antigen fragment. CD4 binds to that. Antigen is presented. The T cell will go on to have its immunological response. MHC class 1 comes from an antigen presenting cell. Technically can come from any antigen presenting cell that has a nucleus, any nucleated cell, and a cytokine T cell binds to that MHC class 1, reads the antigen fragment, says, oh wow, okay, I'm gonna go look for this. It goes and looks for it and kills it. Notice in both cases you need a secondary receptor, CD80 and CD28, CD80 and CD28 to also bind. A complex process that is not fully understood occurs after binding, when activation of T cells by antigen sequesters two signals. Binding of the antigen within the lymphocyte receptor initiates a signaling cascade which involves other membrane-bound proteins and intracellular messengers. When a second signal is communicated along with the antigen, lymphocyte proliferation, differentiation, and expression of specific cytokine genes also occurs. So they're saying that something's not well understood is that, well, yeah, this happens, but what happens afterwards? Some crazy stuff happens. This also leads to effects in the cell, and that's what's not understood so well, is what happens in here. But we do know that bonding of an antigen within the lymphocyte receptor initiates a signal cascade which involves other membrane-bound proteins and intracellular messengers. And when a second signal is communicated along with the antigen, lymphocyte proliferation, differentiation, and expression of specific cytokine genes also can occur. Here's superantigens as mentioned on the test cram I did. Several bacterial and viral proteins can provoke drastic and harmful responses when they are exposed to T cells. And this can occur and be harmful because they stimulate stronger immune responses than normal antigens by tricking T cells into activation, although they have not been triggered by a specific antigen. So these superantigens are, again, there's several bacterial and viral proteins that can provoke drastic and harmful responses. So even though they haven't exactly been activated, they've been tricked into thinking they're activated. And this stimulates T cells to proliferate non-specifically. So they just proliferate like wildfire without even really specifically trying to find anything. So they technically just keep making T cells that do nothing. This contributes to the microbial pathogenicity by stimulating release of massive quantities of cytokines from T cells. This can lead to systemic toxicity and suppression of specific immune responses. Examples of this would be staphylococcal enterotoxin B, which causes food poisoning. It is also on the select agent list as a potential agent of terrorism. The toxin that causes toxic shock syndrome. And mouse tumor virus superantigen. Putative proteins from Epstein-Barr and rabies viruses. These are just examples of superantigens that can cause an effect by tricking T cells into activating, even though they have not been triggered by a specific antigen. They also stimulate T cells to proliferate like wildfire, not specifically. And this could lead to large amounts of cytokines being released. So, tricking activation, high non-specific proliferation, system toxicity due to large amounts of cytokines, and those cytokines can suppress the immune response, the specific immune response. And tumor cells can do this. And I want to say HIV might do something similar, kind of hijacks the T cells, but I would say tumor cells would want to do this more. This would be advantageous to that. And even if you had tumor cells and you had superantigens present from something else, then you're not going to be able to fight that tumor cells much due to the superantigens overactivating your T cells non-specifically and kind of crowding your immune system, causing toxicity and all that. But B cell biology, like T cells, B cells must be activated by a specific antigen to continue mitosis. So they stop dividing and they just kind of go dormant and relax until they're activated by a specific antigen. That's what B cells do. B cells will then replicate and differentiate into plasma cells, which secrete antibodies. They'll also make more memory cells, but that's a side note. The B cells have immunoglobulin receptors for the specific antigen that will activate that particular B cell. So this would be the variable portion of the antibody that has specific antigens, has specificity for the antigen that will activate that B cell. So here's my antigen. If it's right there, just nice and snug, that's going to actually activate this B cell. B cells have immunological receptors for the specific antigen that will be activated. I just said that. These receptors associate with other proteins and are called B cell receptors. So this is the antibody. Anyways, these receptors associate with other proteins and are called B cell receptors. An interaction with that antigen is communicated to the nucleus via a signal transduction pathway similar to that described for T cells. You have signals being transduced into second messengers within the cell, which cause a cellular response to release to go into the nucleus to tell the nucleus to make antibodies, and it's going to make those antibodies and it's going to release them from the cell. You also have these secondary proteins, immunoglobulin A, immunoglobulin beta. So Ig alpha and Ig beta would help be secondary sources of signals for secondary responses inside the cell. B cell biology continued. We're talking about immunological roles. So what do? They proliferate and differentiate into plasma cells and memory cells, which respond to antigens by producing antibodies. And memory cells. I already said that. Internalize antigen. They internalize the antigen and act as antigen-presenting cells. Yep. B cell activation leads to proliferation and differentiation into plasma cells. I keep saying that. And memory cells. But when they become plasma cells, some cytokines are produced by helper T cells that can act on a B cell and assist in growth and differentiation. So T cells can essentially say, hey, you're doing a good job, keep doing what you're doing, keep it up, grow, differentiate. And those are, I forget what kind of T cells. I think it's the ones that are associated with humoral, which were maybe it's TH1 or something like that. B cell activation is typically antigen specific, so the antigen obviously has to bind to the antibody, and that there has to be an antigen antibody complex forming for B cells to be activated. Or there can be T dependent where T cell comes in and activates a B cell with cytokines. So T dependent would be with 2 cells activating the B cell with cytokines. T independent would just be where the antigen comes in and bonds to the antibody. So T dependent, like T cells, these require 2 signals. Antigen, BCR, specific interactions. So this is just antigen B cell receptor, specific interactions with an antibody. So this is like antigen antibody specific interactions. And activated T helper 2, so it is 2, not 1. So activated T helper 2 bonds to the B cell presented. That should be presents. So the activated T helper 2 cell bonds to the B cell presented antigen. Oh, so that is presented. Okay. So activated T helper 2 binds to the B cell presented antigen, because B cells are APCs. They will present their antigen to the T cell. The T cell will bind to that little receptor, and it has its own little antigen receptor. Should be a T cell receptor. So this BCR binds to the T cell receptor and secretes B cell growth factors. So these would be cytokines. So it would release cytokines that say, grow, 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 you're doing great. And the B cell says, okay, I can do that. And after that, the B cell will differentiate into a plasma cell and a memory cell. When T-dependent antigen triggering of a B cell, so the same thing. Here's an actual diagram of what I was drawing. 3 cells are involved: macrophages, T helper cells, and B cells, specific for an antigen. So let's say a macrophage comes up to a naive T helper cell that doesn't know anything about what to look for. So it's naive, it's non-active, it's just sleeping. An MHC class 2 molecule will come in, it will bind to a CD4 receptor of a T helper cell, and the CD28 will associate with that B7 over here. And TCR will align with the MHC class 2 molecule. Antigen fragment will be presented. The naive helper T cell will go through clonal selection, and you'll get a activated T helper cell, TH2, which will go on to activate a B cell by presenting an antigen to that B cell. When it does that, it shoves it into the specific pocket of the B cell. And these probabilities are crazy, how specific all of this is. But this activated TH2, T helper cell variant 2, will present its antigen to the B cell. This will cause a cellular response where the B cell will proliferate, and it will create memory cells and plasma cells. That's a plasma cell. So you can get a memory cell being made. It proliferates, becomes, it's not saying the difference, but you can get a memory cell, a clonal, just proliferation. So it proliferates. Some of these go off to be memory cells, some of these go off to be plasma cells. But this one would grow and differentiate into a plasma cell. And this one may grow and differentiate into another B cell. This one, another one may go off and make a plasma cell. A plasma cell, copy cell, a clone cell, and a plasma cell. Memory cell, that could go off and be a memory cell. This can go off and be another B cell. This can go off and be a plasma cell. This plasma cell is larger. It's going to be circulating in the blood, and it's going to be useful to sit there and wait for. Actually, it's not even going to wait. These plasma cells are active. They're active, and they're actually the main producers of antibodies. They go crazy. See all this? It shows what 1, 2, 3, 4, 5, 6 here. There actually should be like millions of these little things shooting all over the place. So these guys actually go nuts. So that's what this is. This is just T dependent. So the activation of the B cell here is dependent on a T cell. And that T cell had to be activated itself in order to go present to a. So you technically have a T cell presenting to an antigen presenting cell, which is very strange, but that's what happens. So in T-independent antigen triggering, you have T-independent antigens, where polymeric antigens with large number of identical epitopes, like polymeric, it's just a polymer antigen with a lot of identical epitopes. So this would be bacterial lipopolysaccharides, or maybe like the tail of a flagella right here, maybe. And but mostly like lipopolysaccharides of I want to say gram-negative bacteria. These are less effective than T-dependent B cell activation. Less effective than T-dependent B cell activation. Which makes sense because it's not as specific. It's not as amazing, and honestly, it's more random. So this means the antibodies produced have a low affinity for the antigen, and no memory B cells are formed. So T independent, no memory cells. In T dependent, you do get memory cells being formed. And in T independent, you have antibodies produced that have a low affinity for the antigen. They're less specific. Okay. Antibodies. Let's get to antibodies. Let's get trucking for this shit. It's already what, like 6 o'clock? Yep. I have to. An hour and 55 minutes of my test. Still gonna post these though. The antibodies. An antibody is immunoglobulin. Is immunoglobulin a glycoprotein made of activated B cells, plasma cells? So yeah, antibodies are immunoglobulins, true. They are glycoproteins that are made by activated B cells, that is true. And they serve as antigen receptors, B cell receptors on the B cell surface, true. These are found on blood serum, tissue fluids, and mucosal surfaces of vertebrate animals. An antibody can recognize and bind an antigen that caused its production. Yes. So when an antibody binds to an antigen, an antibody will then go out if it's just floating around. So let's say you have an antibody just floating around in the fluid, the humor of the cell, under the organism. You'll have this going out and finding that bacteria, and then it'll surround the bacteria with these antibodies. And these antibodies can recognize and bind to the antigen that caused its production. So that's part of why you can get opsonization. Oh my god. So immunoglobulin structure, let's talk about that. all immunoglobulin molecules have the same basic structure. They have 4 polypeptide chains. So they're kind of like this. They have 2 identical heavy chains. They have 2 identical light chains. They have heavy and light chains connected to each other by disulfide bonds. And both chains contain 2 different regions. Constant regions and variable regions. So constant, variable. And notice that this right here is also constant, and this right here is constant. So the constant region on both the heavy and light chains, and there's a variable region on both the heavy and light chains. Yeah, clean that up. Be seen a little better. So yeah, I'd actually kind of like halfway through from here down on this heavy chain would be constant. From here down on that light chain would be constant. From here down would be constant. From here up around this area on both chains, variable. And this is actually where the antibody comes and binds. I mean, the antigen comes and binds is right here. Both chains contain 2 different regions. That's constant light, constant heavy, variable light, variable heavy. So here we have where the antigen comes in and binds. Disulfide bridges, disulfide bonds connecting to crystallizable fragments, FC. Be right here. FC here. Those are also the constant regions. You see what I mean by that? Pretty self-explanatory. It's not that hard. This is the hinge region. I don't think you have to know that. But this is a different. Holy shit. Okay, so just a little bit different antibody structure. You have your variable heavy, constant heavy, constant light, variable light, constant light, variable heavy, constant heavy. Disulfide bridges all throughout here. Constant and variable domains, hyper variable regions right here. These are what give specificity to binding to antigens. The major antibody in the human serum is the IgG. I get going. It gets going across the placental membrane. But 80% of serum immunoglobulin or the major antibody in human serum is immunoglobulin G. 80% of serum immunoglobulin is used for opsonization, neutralization, activates complement only immunoglobulin that can cross the placental membrane for natural passive immunity to a neonate or a little baby. IgD is abundant on the surface of B cells. So IgD is really abundant on surface of B cells. It is part of the B cell receptor complex. So it's a BCR. Signals B cells to start antibody production. So write that down. IgD, abundant on the surface of B cells. It's kind of curved, but you see the same basic structure here. It is part of the B cell receptor complex, BCR complex, and it signals B cells to start antibody production. So IgM, this is what I call I get mad, because look how crazy it is. It does not leave the blood or cross the placenta. It enhances phagocytosis of target cells, because it gets mad and it starts eating everything. And up to 5% may be hexameric agglutination. Agglutination activates the complement. So these can be hexameric, and they mainly don't leave the blood or across the placenta. They're pretty big. They usually occur as hexameric, or actually pentameric IgM. They mostly occur as that, but they can be hexameric. 1, 2, 3, 4, 5. So this is 1, 2, 3, 4, 5 pentameric, but some like 5% may be hexameric. Agglutination activates the complement. So these are mainly just used to enhance phagocytosis of a target cell. IgA, Immunoglobulin A. Whoa. They're linked by a secretory component across the J chain. Basically, all it's saying about this is it's a dimer. It's a secretory immunoglobulin A. And monomers and dimers secreted across the mucosal surfaces. Tears, you can find them in tears, saliva, breast milk, malt. Tears, saliva, breast milk. I know, I'm not sure. And malt. Okay. Immunoglobulin E is the lowest immunoglobulin serum level, so it's the lowest one that you can find, and is elevated in parasitic infection and allergic reactions. So it sounds like this is actually important to eosinophils, but I don't want to say that. It's too much to say. But opsonization, it binds to receptors on dendritic cells and macrophages. And mast cells will actually find the FC portion. This portion. I went to that. So it actually finds this portion right here and binds to it and are activated to degranulate vasoactive granules when that Fab portion binds allergens. Bind an allergen. to the Fab portion. Mast cells will bind to the Fc portion and are activated to degranulate vasoactive granules. So degranulate vasoactive granules, like I guess histamine stuff, when an allergen would bind. So the kinetics of antibodies, antibody synthesis and secretion can also be evaluated as a function of time. Monomeric IgM, so that's the only one that can. This is the weird pentameric one. So monomeric IgM is the B cell receptor for antigen, whereas B cell activation, pentameric IgM is secreted. So when it's activated, pentameric IgM is secreted right here, but it's just normal IgM before that. So this is called class switching, where a change in antibody class secreted by plasma cells under the influence of T helper cells, which are responding to alternative stimuli, even unfolds with time. So primary antibody response, several day latent periods after initial exposure to the antigen. You get no antibody detectable in the blood. And after B cell differentiation into plasma cells, antibodies are secreted, and millions get secreted. An antibody titer is used and is a measure of a serum antibody concentration and reciprocal of the highest dilution of antiserum that gives positive reaction. Antibody titer is a measure of the serum antibody concentration. Reciprocal of the highest dilution of antiserum that gives positive reaction. So it just shows you, it allows you to track the concentration of antibody being produced, and that tells you how much activity you're getting from the primary antibody response from a B cell. So in a primary antibody response, IgM appears first, followed by Ige glad. So you get mad. So I get mad before I get glad. That's the primary. So here we have antibody production and kinetics, crazy shit going on, but primary response, you have your latent period, your log phase, your total antibody, plateau phase, decline phase. You'll have your latent period, your IgM, and then it dies off quick. Then you have a really long latent period for IgG, comes up and dies off later. In a secondary response, your total antibody, boom, like that. You have your IgM, you get mad first, then you get glad. So in both cases you get mad and glad, but Secondary is a little different. It doesn't last as long as the primary response until. So pretty much the activity kinetic shows up later in the primary response than the secondary response. So this means that you actually, and you don't get the decline phases really showing for the total antibody like you would for primary response. You can, the primary response, you're actually going to get your primary response to that antigen coming in. So it's annuity. You got to post your wanted signs. Here, wanted signs posted, boom, done, boom, done, boom, done, all before the whole 20 days or so are over. So the first exposure to an antigen, your B cells split off. IgM, then IgG occurs. IgG occurs, it goes away, you get your plasma cells. You have memory cells that just keep going the whole time. And these all die off. That's what this is. You have memory cells just there. Maybe like 2. You have secondary exposure to an antigen. You have your IgM appear. You get mad. You start getting glad. You get really glad. You get mad. You get really glad. So you're really glad. You have more of an IgG response this time, and a secondary response in reference to a primary response. You have a lot more plasma cells being made, a lot more memory cells being made. So with each secondary subsequent exposure, you have more and more memory cells. So the more you expose yourself to an antigen, the more likely you are to combat that. This is why a lot of people are immune to it, that have slow exposure to certain types of snake venom. Actually have so many memory cells that they can actually just inject snake venom into themselves and they won't have to worry about it. But the secondary antibody response, upon a secondary exposure to the same antigen, B cells mount a heightened or anamnestic response. This is characterized as having a shorter lag, a more rapid log phase. So the latent period, not as long, it's about 5 days versus like 10. It's actually less than 5 days. A more rapid log phase. So look at this log phase. Boom. Increases really quickly in all these cases. Longer persistence. Notice how long they last. They don't really die off as quickly. So there's like 10 to like 20 days. Only lasted 10 days. This would be like 5, and then 20 days goes and still going strong. So there's longer persistence. Higher IgG titer. So yeah, it titers. It hits at a higher point on the IgG. And production of antibodies with a higher affinity for the antigen. Diversity of antibodies. There's 3 mechanisms that can contribute to the generation of antibody diversity. You can get rearrangement of antibody gene segments, gene shuffling and I believe that's in the variable regions. These genes are split or interrupted into many gene segments. Generation of different codons during antibody gene splicing. So yeah, you get a generation of different codons during antibody gene splicing. And you get somatic mutations. So you can get mutations, gene shuffling, and generation of different codons during antibody gene splicing. So in light chain production, you're going to create saying that B cell DNA, if your germline DNA for light chain that has multiple coding sequences, V and J joining in the B cell development, one V is joined with one J region. Many possible combinations are formed. VJ joined with C constant exon. So they spliced out what they didn't need, and they kept with. So this is basically just deleting and intervening. So J4 was deleted out, and then this came in. So transcription, you get splicing and translation. You have an intron being made. His intron is excised, and then you have translation, and then boom, it gets turned into the light chain with missing one of these, which creates variability in the light chain. So in a heavy chain production, you have, your V2, D2, J3, all these C's, and J3 and 4. So J3 and 4 come in. You have another intron. Intron gets spliced out. Boom. And they kicked out 4. Now you have V2, D2, J3, and C beta. Seems like that got cut out and that got cut out. But then you have this, and the B cell DNA after a second DNA splice. So it goes back in, it takes this, splices it, adds in something else, and then adds in something else. So like you really have this going on, then you have this going on and forming with this. So you just have second DNA splicing, and then all of a sudden you have the intron taken out and these combine, and now you have this with this replaced with that. So it's pretty advanced, but that's what essentially goes on. It's introns and exons. Exons just keep getting linked together because they are the coding regions. So V and J regions are joined to the third coding region called D diversity sequence. VDJ joined to a C region. Antibody class switch, class beta to class Y. And then you get initial C region results in an IgM, but changes as the immune response progresses and B cells proliferate. So you have your germline DNA. This explains how you can go from a germline DNA where you have IgM, and then you all of a sudden end up with, boom, IgG. What does it do? I'm going to take a screenshot of this. Please work with me. Okay, apparently my phone's retarded right now. And it refuses to take a screenshot. Okay. fucking pissed me off. Anyways, I'm gonna keep going where I'm going. Fuck that screenshot. So with what looks like gamma light chains, B region is 2, J region is 3, 2 times 3 is 6. So 250 to 350 variable K regions is what they're saying. And JK regions, 4. So 250 times 4 is 1,000, and 350 times 4 is 1,400. So they have 1,000 to 1,400 light chain combinations here. So here in the heavy chains, we have 250 with 1,000 with 10 to 30. 4, so you get 250, 10, 4, 1,000, 30, 4. So you have 10,000 to 120,000. And then to see how many K containing antibodies you have 1,000. 1,000 times 10,000 and 1,400 times 120,000. That's ridiculous. The alpha containing ones. Six times that and 6 times that. Yeah, whatever. Clonal selection theory. Body forms large, diverse beta lymphocyte or B lymphocyte pool. Clonal selection theory says that a body forms a large, diverse B lymphocyte pool that can bind to large range of antigenic epitopes. The body forms a large, diverse B lymphocyte pool that can bind to a large range of antigenic epitopes. There are self-reactive cells that are eliminated at an early stage of development, clonal deletion. These are self-reactive cells. Wake up for a second. The clonal selection theory body forms a large, diverse B lymphocyte pool that can bind to large range of antigenic epitopes. So the body forms a large, diverse B lymphocyte pool that will then bind to a large range of antigenic epitopes. These are self-reactive cells that are eliminated at an early stage of development, clonal deletion. Okay, so basically clonal selection theory says that the body forms a lot of diverse B lymphocyte or B cell, a bunch of big ass diverse B cells that combine to a shitload of antigens, and any self-reactive cells are thrown out in the early stages of development because you don't want to be reacting with yourself. And the encounter with antigens stimulates only the B cells that recognize and bind that antigen. So only those that can bind to that antigen will be stimulated, and the stimulated B cells will proliferate to produce B cell clones and memory cells. All have the same antigen specificity. It depends on if it makes memory cells or not, whether or not it's T cell specific or T cell dependent or T cell independent. T cell independent does not make memory cells, although T cell dependent does. Stimulated B cells proliferate to produce B cell clones and all have the same antigen specificity. B cell clones differentiate to form two cell populations, plasma cells and memory B cells. Holy balls, Batman. During development of early lymphocytes from stem cells, a given stem cell undergoes rapid cell division to form numerous progeny. During this period of cell differentiation, random rearrangements of the genes that code for cell surface proteins and receptors occur. The result is a large array of genetically distinct cells called clones. Each clone bears a different receptor that is specific to react with only a single type of foreign molecular antigen. At the same time, any lymphocyte clones that develop a specificity for self molecules and can be harmful are eliminated from the pool of diversity. So these guys that bind the self, these guys bind the self, are killed off. They go, they won't make it. At the same time, any lymphocyte clones that develop a specificity this is called immune tolerance when these guys die. The specificity for a single antigen molecule is programmed into the lymphocyte. It is set for the life of a given clone. The end result is an enormous pool of mature but naive lymphocytes. They're inactive and they're not really... they're inactive, waiting to be activated, and that are ready to further differentiate under the influence of certain organs and immune stimuli. Antigen dependent period. So this is when they're not dependent on antigens. They don't have an antigen binding to receptors yet. They're still differentiating and going through the process of immune tolerance and immune tolerance differentiation, and then they become naive cells, waiting to be simulated. But during the antigen-dependent period, lymphocytes come to populate lymphatic organs. There they will finally encounter antigens. These antigens will become the stimulus for the lymphocytes' final activation and immune function. Entry of a specific antigen selects only the lymphocyte clone or clones that carry matching surface receptors to the antigen antibody interaction. This will trigger an immune response. So once the antigen bonds to the antibody, there will be immune response, which varies according to the type of lymphocyte involved. So, action of antibodies. These bind antigens with great specificity can occur in animal body in vivo, can also occur outside the body. They are essential for the protection of animals from viruses, microbes, and cancer cells. Antibodies coat foreign invading material. This is opsonization. It marks it for recognition by components of the innate and adaptive immune systems. Antibodies can undergo neutralization, opsonization, and immune complex formation. Consequences of antigen-antibody binding. Precipitation. You can have antibody antigen complexes forming and causing precipitates to form. So you can get this actually happens when you give someone the wrong blood. They coagulate like that. You get precipitates forming in the blood, and they can clot them up and kill them and stop their heart and all kinds of stuff. It's really bad. So cell-free molecule in solution, antigen, and antibody just kind of meet with each other. Opsonization, you surround the bacteria with antibodies to be engulfed with a macrophage. You have neutralization, antibodies block binding. So these will be like IgM antibodies would be very good for blocking, neutralizing viruses. And these are actually 1, 2, 3, 4, 5, 6. So those are hexameric, I think. Agglutination would be where cross-linked bacterial cell antigens just kind of cross-link. So actually in the blood, this is really what happens. You have this happening, and antigen antibody complexes forming on a large scale, and you get agglutination. But on small cells, kind of like agglutination of a small antigen fragment with an antibody. That's called precipitation. Agglutination and precipitation are really similar, but agglutination is when entire organisms connect each other, and precipitation is just when cell-free molecules bind with antibodies. Complement fixation occurs when complement binds to a cell and causes lysing of bacterial cells. Similar to opsonization, except it ends up with lysis instead of phagocytosis. And it requires complements. Toxin neutralization, inactivation of toxins resulting from interactions between toxin and specific antitoxin antibodies. So these antitoxin antibodies actually inactivate the toxins of the invading bacteria by surrounding it and keeping these toxins from being released. Complexing toxin with antibodies can prevent the toxin from attaching the host cells and can prevent toxin from entering host cells. Can result in ingestion by macrophages. So macrophages can come in and clean up the toxins when these antibodies will actually surround the toxin. Say this is a toxin and an antibody surround it. Macrophages come in and eat that. So there's also viral neutralization. IgG, IgM, and IgA antibodies can bind to some viruses and inactivate them. Good ass motherfucking neutralization. That's what I'm gonna say. Good ass motherfucking viral neutralization. Fixation of complement component C3b from the classical complement pathway helps in the neutralization process of viral neutralization. Viral infection is prevented because neutralization of viruses prevents them from binding and entering target cells. Viral infection is prevented because neutralization of viruses prevents them from binding and entering target cells. So viral infection is prevented because neutralization of viruses prevents them from binding and entering target cells. Blah. Let's just start from here because my brain's farting. Fixation of complement component C3b from classical complement pathway, that's true, helps in the neutralization process. Okay. And the viral infection is prevented because neutralization of the virus prevents them from binding and entering target cells. Duh. If you neutralize them, they can't get in the cell and they can't infect the cell. Opsonization, microorganisms or other foreign particles become coated with antibodies and or complement. Opsonizing antibodies bind the Fc receptors, the constant region, on the macrophages. Opsonizing antibodies bind Fc receptors on macrophages and neutrophils. So on their antibodies, I guess, receptors, antigen receptors. Opsonizing antibodies bind Fc receptors on macrophages and neutrophils, creating a bridge between the phagocyte and the antigen. Oh, so Fc receptors bind the opsonizing antibodies on macrophages and neutrophils, making a bridge between phagocyte and antigen so that phagocytosis can occur. Immune complex formation. Antigens and antibodies can cross-link, producing immune complexes. Oh, man. Antigens and antibodies can cross-link, producing immune complexes. Precipitation reactions occurs when antigens are soluble molecules and the immune complex settles out of solution. Oh, so otherwise soluble molecules will precipitate out when antibodies are present to bind in that antigen. So you might have, well, if you had an antibody that could bind to this particular lipid or something that was just soluble somehow, if it were to, or some kind of molecule that was soluble, like say you had a, for example, just one that just wildly was able to bond to sodium. You throw salt in a solution, and then you throw that antibody in, what's going to precipitate out, and you'll have a sodium antibody precipitate formed. Immune complex formation. Agglutination reactions occur when cells or particles are cross-linked. The immune complex formed is more readily phagocytized in vivo than are free agents. This is caused by agglutinin antibodies. So, immune complex formed is more readily phagocytized inside of an organism than are free agents. That's because they kind of clump together so macrophages can come in and suck everything up. This is caused by agglutinin antibodies. Antibody-antigen ratio is equivalent zone when their concentration is optimal for formation of the immune complex. Okay, so when the immune complex formation occurs, you get agglutination. This is generally when you're at the equivalent zone where the ratio between the antibody and antigen is the same. Is the basis for many immunological assays. Summary: the role of antibodies and lymphocytes in resistance. Resistance to viral infections involves both humoral and cell-mediated immunity, as well as interferon sensitization of host cells. Resistance to bacterial infections provided primarily by the humoral immunity, which releases antibodies. So resistance with viral infections involves both the humoral and cell-mediated immunity, as well as interferon sensitization of host cells. Resistance to bacterial infections provided primarily by the humoral immunity. So if you have a bacterial infection, most of the resistance to that antigen, bacterial antigen, is going to be by your humoral immunity creating antibodies. But if you have a viral infection, you're going to be using humoral and cell-mediated immunity, as well as interferon sensitization. Resistance to viral infections. Antibodies neutralize viruses. Antibodies enhance phagocytosis of viruses by opsonization, and they interfere and cause death of infected cells. Some interferons stimulate activity of T cells, and CTLs kill virus-infected cells. CTLs kill virus-infected cells, true, in natural killer cells, and they also kill tumors and cancer cells. NK cells stimulated by interferons and antibodies. Cytotoxic T lymphocytes destroy virus-infected cells and are also involved in the destruction of cancer cells by a process known as immune surveillance. Cytotoxic T lymphocytes destroy virus-infected cells and are also involved in the destruction of cancer cells by a process known as immune surveillance. Resistance to bacterial infections. Certain antibodies and complement proteins act as opsonins. IgM and I get mad and I get glad, agglutinate bacteria. Antibodies trigger formation of membrane attack complex. Certain complement proteins attract phagocytic cells. That's true. Antitoxin antibodies neutralize bacterial and exotoxins. Cytokines attract and stimulate macrophages. Yeah. And cytokines also stimulate T cells and increase populations of cells involved in cell-mediated responses. So T cells can secrete cytokines to tell B cells to proliferate and increase their proliferation and differentiation. Immune disorders, you can have hypersensitivities, autoimmune disease, transplantation, tissue rejection, and immunodeficiencies. Hypersensitivities, you have exaggerated immune response upon second or subsequent contact with antigen, so you have too much of immune response. You might have too much inflammation, so there's a high chance with people actually that have themselves mount a response to snake venom all the time. At any point, you can become allergic to it instead of immune, because you can have a chain reaction occur where you have all of those immune cells, causing inflammation and all kinds of stuff. And then it would be bad to actually be hypersensitive. In that case, it can cause tissue damage because of inflammation. It'd be like chronic inflammation in a way. Reactions are classified as immediate or delayed. And Jill-Coombs classification gives 4 different types: 1, 2, 3, and 4. Type 1 hypersensitivity, or like an allergy. One kind of type 1 hypersensitivity. An allergen, antigens that cause allergic reactions, occurs immediately following second contact with the allergen. It involves production and action of IgE, sometimes called reagin, and mast cells, and sometimes basophils or eosinophils. So with the environment, you generally come up with IgE, typically involved with mast cells, basophils, and eosinophils with allergies and sensitivity and hypersensitivity. That'd be IgE. So here we have sensitization of Ige production. Allergen particles enter. You have a mucosal membrane, lymphatic vessel. You have your pollen come in, bind to a B cell. B cell recognizes the allergen with help of the T cell. This proliferates into plasma cells, which synthesize IgE. And IgE goes on and binds to mast cell surface receptors and mast cells in the tissue. Primed with IgE will then attach to the allergen, which stimulates degranulation, which releases allergic mediators. And you have systemic distribution of mediators into the bloodstream, and you end up with red, itchy eyes, hives, runny nose, fever, swelling, pain. So this is how you can get IgE attaching to mast cells. The combination of these antibodies with allergens then stimulates the mast cell or basophil to degranulate and produce the physiological mediators that cause the anaphylactic reaction of inflammation and asthma or hay fever. So anaphylaxis is the release of physiological mediators in response to allergen cause. Smooth muscle contraction, vasodilation, increased vascular permeability, mucus secretion, can be systemic or localized. So anaphylaxis means the release of physiological mediators in response to allergen cause. Yeah. Smooth muscle contraction, you end up with vasodilation, increased vascular permeability, mucus secretion, and can be systemic or localized. So anaphylaxis is literally just releasing of physiological mediators in response to an allergen like pollen, where you get smooth muscle contraction, vasodilation, increased vascular permeability, mucus secretion, can be systemic or localized. It's like an allergic reaction. You can have systemic anaphylaxis, results from the massive release of mast cells mediators in a short period of time, which can be bad. It usually results in respiratory impairment. You get inflammation of the lungs, decreased blood pressure, circulatory shock, and could cause death within a few minutes. These would be people that have EpiPens laying around from when they're allergic to things. Yeah, this would be anaphylactic shock when you die. Localized anaphylaxis is just an atopic, out-of-place reaction. Symptoms depend on route by which the allergen enters the body. So, like, this would be like hay fever, you have an upper respiratory tract reaction, which I've had before. I've had that plenty. Bronchial asthma, lower respiratory tract, anaphylaxis, hives. I mean, I haven't had essentially hay fever, but I've had a lot of upper respiratory problems that I don't have anymore. Thank God. Bronchial asthma is a lower respiratory tract. Hives, you get hives on the skin. Common with true food allergies. Diagnosis and treatment. Diagnosed by skin tests. Inoculation of small amounts of allergens into the skin. Rapid inflammatory reaction characterized by redness, swelling, and itching. Desensitization, controlled exposure to an allergen, stimulates immunoglobulin Ige production, and intercepts and neutralizes the allergen before it binds to IgE-bound mast cells. So desensitization will be when you control exposure to the allergen and stimulates IgG production, which will then intercept and neutralize the allergen before it binds to the IgE-bound mast cells and causes the little mediators to be released that would cause inflammation, which you don't want. Diagnosis and treatment, diagnosed by skin tests, inoculation of small amounts of allergens into the skin to see how it reacts, rapid inflammatory reaction characterized by redness, swelling, and itching. Like this. Allergy testing to see what they're allergic to. Obviously allergic here. Type 2 hypersensitivity, cytolytic or cytotoxic reaction. Involves IgG and IgM antibodies are directed against cell surface or tissue antigens, stimulate complement pathways and effector cells. Type 2 hypersensitivity, IgG can stimulate NK cells, platelets, neutrophils, and macrophage monocytes and effector cells that can cause plasma membrane damage and cell death. IgM and IgG, C1q, classical pathway, lytic pathway, plasma membrane damage and cell death, activated C3. The activation of an antibody occurs through effector cells or the membrane attack complex of complement, damaging the target cell plasma membranes, causing cell destruction. Blood transfusion reaction. Agglutination, and when you get agglutination occurring because antibody antigen complexes form within the blood. Erythroblasts fetalis, the mother can be passively immunized with an anti-Rh factor, antibodies, or ROGAM to control this disease, which is potentially fatal in newborn. It usually can cause jaundice in some cases if it's not severe. It happened to my sister. Yeah, so the mother is like, there can be like some placental leaking across. And then you get, you get reactions like this in the blood. Type 3 hypersensitivity involves formation of immune complexes, usually removed by monocytes and macrophages. If accumulate, leads to hypersensitivity reaction. Resulting inflammation causes tissue damage, vasculitis, glomerulonephritis, arthritis, and systemic lupus. Basically, antibody combines with excessive soluble antigen, forming large quantities of antigen-antibody complexes. Circulating immune complexes become lodged in the basal membrane of epithelial sites such as kidney, lungs, joint, skin. Sounds like jaundice to me. Fragments of complement cause release of histamine and other mediator substances. So neutrophils migrate to the site of immune complex deposition and release the enzymes that cause severe damage to tissues and organs involved. So top 3, you get formation of immune complexes, usually removed by monocytes and macrophages. If accumulate, leads to hypersensitivity reaction. Resulting inflammation causes tissue damage. So you have tissue damage in type 3. So you have tissue damage because of a lot of antigen antibody complexes forming from soluble antigens forming large quantities of antigen antibody complexes. Type 4 involves delayed cell-mediated immune reactions. Important factor is time required for T cells to reach and accumulate near antigens. TH and CDL cells have elicit type 4 reactions. TH and CDL cells can elicit type 4 reactions. Try to cover this faster. I'm getting tired. 50 minutes to my test too. Need to review some stuff. Delayed type hypersensitivity. Tuberculin skin test. They slap some of that into you. Contact dermatitis. For example, poison ivy. You have chemical antigens. Yada yada. Th1 tumor necrosis factor. Interferon. Memory T helper cell. Blood vessel. Macrophage. Induce inflammatory reaction. Inflammatory fluid. Kill skin cells with cytotoxic C cell. So they put lipid-soluble catechol into the skin and dendritic cells, close the epithelium, pick up the allergen, process it. Th1 CD4 cells recognize and present that allergen to your macrophage. Sensitized Th1 cells are activated and secrete cytokines. These cytokines attract macrophages and, cytotoxic T cells to the cell. So, yeah. Macrophages release mediators that stimulate a strong local inflammatory reaction. Cytotoxic C cells kill cells damaged. You get blisters like this. Autoimmune diseases, autoimmunity, presence of serum antibodies that react with self-antigens, often benign. Naturally, consequence of aging because you have more genetic errors as you age. So reversibility induced by numerous stimuli, infectious organisms or drugs or chemical exposure, I'm sure. Autoimmune disease that results from activation of self-reactive T and B cells and leads to tissue damage. Rheumatoid arthritis, insulin-dependent diabetes mellitus, effects that influence the development of autoimmune disease. You have genetic, viral, hormones, and an influence of stress and neurochemical on the immune response. Transplantation tissue reduction. These are types of transplants. We have allograft where Transplants between genetically different individuals within a species occur. A xenograft where donor and recipients are different species. I think out of the same species graft between two genetically different individuals of the same species. A xenograft is a donor and recipient that are different species. Two mechanisms can occur. Foreign MHC proteins molecules on the transplanted tissue are recognized by the host helper T cells which then aids cytotoxic T cells in destroying the graft. The T helper cells react to the graft by releasing cytokines, which stimulate destruction of the graft by macrophages. Tissue transplantation to be successful, the ABO blood group and the MHC molecules of the donor and recipient must be as closely matched as possible, and after matching blood types of donor and recipient, this identification of human leukocyte antigens is done. Family members are the first choice for a close match because HLA genes are usually inherited as a complete set from the parents. Graft versus host disease can occur in an organ transplant recipient. Immunocompetent cells in a donor tissue reject the host, like bone marrow transplants. Disease prevented by treating donors with immunosuppressive drugs to help deplete marrow of mature T cells. Immunodeficiencies, failure to recognize and respond properly to foreign antigens. Primary congenital immunodeficiencies results from a genetic disorder, and acquired immunodeficiencies, which results from infection by immunosuppressive microbes like HIV that would stop T cells, or not just specifically T cells, but tumors will stop T cells. Oh, I think it's over. Bye-bye.